Venom, toxic compounds injected into another organism, has evolved independently in at least 20 animal lineages. The diversity of mechanisms, targets, and delivery systems challenges any simple definition of what venom is for. In several cases, the most lethal venom belongs to the least aggressive animal, and the deadliest bites are indistinguishable from harmless contact until it is too late. The inland taipan, Oxyuranus microlepidotus, is the most venomous snake on Earth by subcutaneous LD50, the measure of venom lethality per unit of body weight in test animals. A single bite delivers up to 110 milligrams of venom, sufficient to kill approximately 250,000 mice or, theoretically, 100 adult humans. The venom contains a cocktail of neurotoxins, hemotoxins, and myotoxins that work simultaneously, paralyzing the nervous system, preventing blood from clotting, and destroying muscle tissue. Despite this, the inland taipan has never been confirmed to have killed a human being in the wild. It is reclusive, inhabits remote Australian outback regions, and consistently attempts to retreat before striking. The handful of recorded bites have all been to snake handlers who provoked the animal at close range. All received antivenom and survived. Blue-ringed octopuses, Hapaloclina lunulata and related species, weigh approximately 25 grams, roughly the size of a golf ball, and carry enough venom to kill 26 adult humans. The venom is tetrodotoxin, TTX, one of the most potent non-protein toxins known. It blocks sodium channels in nerve cell membranes, preventing nerve signals from firing, causing rapid paralysis of voluntary and involuntary muscles, including the diaphragm. Death from respiratory failure can occur within minutes. There is no antivenom. The only treatment is sustained artificial ventilation until the toxin clears from the system, which takes approximately 24 hours if the victim survives the initial respiratory arrest. The bite is painless. The first symptom a victim typically notices is numbness, followed by difficulty breathing. The blue rings appear only when the animal is threatened. At rest it is brown and camouflaged. Box jellyfish, Chironex fleckeri, are considered the most acutely lethal animals on Earth by speed of kill. Their tentacles, up to 3 meters long, with 5,000 stinging cells per square centimeter, contain a venom that attacks the cardiovascular system directly, causing cardiac arrest within 2 to 5 minutes of a significant sting. The venom contains a pore-forming toxin that punches holes in red blood cell membranes and destabilizes the electrical signals controlling the heart. Victims of severe stings have died before reaching shore. The jellyfish has 24 eyes, arranged in 4 clusters, with corneas, lenses, and retinas, making it one of the few animals that can form true images despite lacking a centralized brain. The connection between the eyes and the stinging cells appears to be direct. There is no neural processing between visual detection of a target and nematocyst discharge. The cone snail, Conus geographus, injects venom through a harpoon-like tooth that extends to any point on its body, allowing it to sting a person holding it from any angle. The venom contains hundreds of distinct peptide compounds, conotoxins, each targeting a specific receptor or ion channel in the nervous system. No 2 cone snail species have the same venom composition. Some variants have been found to block pain receptors so effectively that a synthetic analog, zaconotide, is now used as a painkiller 1,000 times more potent than morphine. Geographic cone snails can deliver enough venom to kill an adult human. Treatment is supportive only, no antivenom exists, and there has been no known recovery from a severe envenomation without intensive medical support. Collectors have died after picking up live cone snails. Slow lorises, Nycticebus sp., are the only venomous primates. They produce a secretion from a gland on their inner elbow. When they lick this gland and mix the secretion with saliva, it becomes toxic. The mechanism of envenomation is the bite itself. The venom causes tissue necrosis at the site, anaphylactic shock in some individuals, and in small mammals, death. Uniquely, the chemical structure of the slow loris brachial gland secretion is similar to that of cat allergens, suggesting either an evolutionary origin from a cat allergen-like protein, or convergent evolution producing similar chemistry. Slow lorises also use the venom for interspecies competition. Females smear it on the top of their infants' heads to deter predators while they forage. They are critically endangered, partly because the exotic pet trade removes their teeth, eliminating the venom delivery mechanism and their primary defense. Platypuses, Ornithorhynchus anagnus, are one of the few venomous mammals. Males have a hollow spur on each hind ankle connected to a venom gland in the thigh. The venom causes immediate, excruciating pain that is resistant to morphine. Even large doses of opioids do not effectively manage platypus venom pain, and the hypersensitivity can persist for months after envenomation. The venom is not used to kill prey. Platypuses eat invertebrates and detect them via electroreception. The spur is used exclusively during competition between males, particularly in mating season. The venom gland is only active during breeding season. Outside this period, males produce no venom. The genes encoding platypus venom appear to have evolved independently from those of reptile venoms, a parallel evolution of the venom delivery mechanism from different genetic origins. The short-tailed shrew, Blurina brevicata, is one of the few venomous mammals outside the monotremes. Its saliva contains Blurina toxin, which it introduces through its teeth while biting. The toxin paralyses prey items, earthworms, beetles, snails, and occasionally mice larger than the shrew itself, without killing them. The shrew then caches the paralyzed prey alive in an underground larder, where they remain incapacitated until consumed. This allows the shrew to maintain a live food supply, critical for an animal with a metabolic rate requiring almost continuous feeding. A short-tailed shrew bitten by laboratory researchers caused localized pain, swelling, and redness lasting several days, confirming functional venom even when the animal is not actively hunting. Komodo dragons, Varanus komodoensis, were long believed to kill through bacterial infection. Their saliva supposedly contained lethal bacteria from scavenging. This was disproven in 2009. Their lower jaw contains venom glands producing a complex that prevents blood from clotting. When a Komodo dragon bites large prey, the anticoagulant venom causes continuous hemorrhage from the wound. Combined with the mechanical damage of the bite, this causes the prey to weaken over hours or days and eventually collapse. The infection hypothesis was incorrect in its mechanism but coincidentally true in its prediction. Animals bitten by Komodo dragons do die, but from venom and blood loss, not from bacteria. Komodo dragons can detect prey via their forked tongues up to 9.5 kilometers away using chemoreception. These venoms represent distinct evolutionary solutions to the same problems: incapacitation, competition, and defense. In several cases, the chemistry they contain has been directly used in human medicine. Tomorrow's fact involves an ability scientists still can't fully explain. This channel posts a new animal fact video every day. Subscribe so you don't miss the next one.